Hi, sir. So I have a question for you. What is pus?
Yes, Mr. David. You have a question, very common question, very basic question. What is pus? Right? Let me explain in the simplest possible way that let's start with the components of pus. Then what are the components of pus? Pus consists of number one, some local cells, local tissue cells, which are alive, dead, and dying.
Okay.
Number two, pus has lot of neutrophils and might have some other leukocytes like macrophages. But most abundant leukocytes which are present in pus is neutrophils. And about the neutrophils, some of the neutrophils are alive, some of them are already dead, and some are dying. So up to now, I've told you two components of pus. That pus in any tissue consists of alive, dead, and dying local cells, plus alive, dead, and dying leukocytes, especially neutrophils. And third component is pyogenic stimulus may be present. The stimulus which produces pus. The most common example we can say pyogenic organism, pyogenic bacteria, pus producing bacteria like streptococcus, pneumococcus, gonococcus, E. coli. There are so many bacteria. Some fungi also produce pus like Aspergillus fumigatus, Cryptococcus neoformans, right? And sometimes there is a foreign body which irritates or some chemical which irritates the tissue and damages the tissue and forces the tissue to produce pus. I will tell you later how pus is produced. Now, so our third component of pus is pyogenic stimulus, right? In case if there are pyogenic organism like suppose staphylococci, some of them will be alive, some of them will be dead, and some of them will be dying.
Yes.
Right? So actually, what is going on there in the tissue with the pus? Let's suppose there's pyogenic organism which has invaded the tissue, and now pyogenic organism is damaging the tissue, and pus is being produced. So that pus will consist of number one, alive, dead, and dying local tissue cells. Number two, alive, dead and dying neutrophils, right? With some other leukocytes. Third, alive, dead, and dying pyogenic microbes, right?
Yes.
And all these things, I mean, local cells, neutrophils, and microbes, all of them are produced and all of them are present in protein rich exudate. All of them are present in protein rich exudate, which makes white or yellowish, creamy, viscous fluid, right? Now, how pus is produced, I will tell you very briefly. I'm not going to teach you whole inflammatory process. I will just tell that why in the pus, number one, why in the pus there is lot of protein rich exudate. The reason being that pyogenic organism damage the tissue and tissue produces a lot of chemical mediators of inflammation, and many of them like histamine or tumor necrotic factor or interleukin 1, these actually act on the endothelial cells, and endothelial cells are activated and they shrink and interendothelial gaps appear. Now, in inflamed tissue, blood flow is, vessels are dilated, there's more blood flow, more hydrostatic pressure, and with that, there is, due to activation and shrinkage of endothelial cells, microcirculation becomes highly permeable, and lot of protein rich fluid moves from vascular compartment to the interstitial compartment. And this fluid, which is moving from the vascular compartment into interstitial compartment, this fluid is being pushed under high hydrostatic pressure plus increased permeability of local microcirculation. A lot of proteins leak out into fluid and proteins leak out into interstitial tissue. So this is the beginning of formation of protein-rich exudate. Secondly, when there are pyogenic organisms, they damage the tissue and some local resident cells like mast cells, resident macrophages, and even injured endothelial cells, all of them are activated and they produce chemical mediators which specially orchestrate the events which lead to the extravasation of specially neutrophils. The neutrophils move from the vascular compartment to the interstitial compartment, right? What happens that endothelium, under the influence of chemical mediators, endothelium become adhesive to the neutrophils. It start expressing molecules like selectins, right? And also selectins and their ligands interact with neutrophils, and neutrophils start rolling into that tissue. Then integrins and their ligands interact with each other. Integrins are expressed on the and activated on the neutrophils, and their ligands like ICAM and CCAM, they are expressed on the endothelial cells. So neutrophils are not washed away through the blood flow from the inflamed area or injured area. Rather, neutrophils stick to the endothelial lining and then through the interendothelial gaps and sometimes even through the endothelial cell, neutrophil extravasate. They come out and then they show once neutrophil have come out of the circulatory system into interstitium, then they have a directional locomotion towards the focus of injury. This locomotion is called chemotaxis. Movement of neutrophils, right? Directional movement of neutrophils towards the focus of injury along the concentration gradient of chemoattractants. And those chemoattractant molecules which specially attract the neutrophils, right, and other white cells, we call them chemokines. And there are many molecules which act as chemokines, but for neutrophil, the most powerful chemokine you should remember, interleukin 8. But of course, leukotrienes B4 also act as a chemokine, and there are other cytokines which act as chemokines. So what really happens? That at the site of injury, where their pyogenic stimulus is present, especially pyogenic organisms, local tissue is damaged, right? When local tissue is damaged, that will produce alive, dead, and dying local cells, parenchymal cells, and stromal cells. Number two, there is accumulation of, due to vascular phenomenon, right? And due to, you can say, leukocyte, especially neutrophil adhesion, and diabetes and chemotaxis, there's accumulation of neutrophils, right? And that neutrophils are fighting with the microbes, and during this war between the microbes, pyogenic microbes, and the neutrophils, this is just like two armies fighting with each other on both sides. Some neutrophil and some microbes are still alive. Some neutrophil and some microbes are injured and dying. Some neutrophils and some microbes, they are undergoing They are already dead, right? And all this is happening in and around the local cells. They are also injured. Local cells are injured by the different destructive enzymes produced by the microbes and also by the destructive enzymes produced by the and or reactive oxygen species products produced by the neutrophil. So what will happen? That at that tissue where army of neutrophils and army of pyogenic microbes are fighting, all this fight is occurring on the local cells or around the local cells. So many local cells are also alive, dead, and dying. And as I told you that lot of interendothelial gaps appear and hydrostatic pressure in the local microcirculation is high and lot of protein-rich fluid is extravasated. Fluid come out of vascular system into interstitium, right? So we say there is a, of course, formation of protein-rich exudate. So let me, in a nutshell, summarize what is pus. Pus is consisting of alive, dead, and dying local cells plus alive, dead, and dying neutrophils, most abundantly. Thirdly, alive, dead, and dying microbe, if there are pyogenic microbes present over there. And all of these alive, dead, and dying cells and microbes, right? All of them are present in protein-rich exudate, right? Which is creamy and viscous and opaque. Remember, pus is opaque, creamy, viscous. And in different organisms, pus might have different colors, right? Now, one question, why pus is opaque, right? Why pus is so viscous? There are multiple reasons for that. Number one, due to lot of tissue debris, due to injured and dead cells, local cells, due to injured and dead neutrophils, due to heavy load of pathogens, plus due to presence of many enzymes, right? Then due to presence of lot of proteins around that area, not only destructive enzymes, right? And also when neutrophils are dying, local cells are dying, lot of DNA is released. This big polymer of polymeric molecules, DNA, right? They also add to the viscosity and opacity of the pus, right? So what can we say that pus is, why it is so opaque, creamy? One reason, there is a lot of proteins, protein-rich exudate, there are dead cells, tissue debris, pathogen loads, and enzymes, and even some macromolecules like DNA is coming out of cells, especially from injured and dead cells, from from neutrophils and from even local injured cells, even from microbes. Now, as I told you, that as far as white blood cells are concerned, the major component of the white cell which is present over there is neutrophil. Usually a question is asked that when there is injury, now I'm coming to another question, because yesterday a student asked me, sir, what is the reason that when there is acute inflammation, why neutrophils are the first to reach at the site of inflammation, right, among the all leukocytes? So I will say, especially when there's pyogenic stimulus, pus forming stimulus, or neutrophil accumulating stimulus, right? Or even simply there is acute inflammation, right? Usually neutrophils are first to respond and accumulate to the site of injury, later on followed by monocytes which come into tissue and become what? Macrophages. And still more later on, lymphocytes will come and other white blood cells will come. Actually, when injury occurs, within first few minutes to up to 6 hours, neutrophils arrive in big waves. And neutrophils keep on accumulating for about 24 hours. Then macrophages start accumulating. And within 48 to 72 hours, neutrophil component in inflammation usually start going down and macrophage components start going up. Question is, why neutrophils are first to come? Why macrophage come later and lymphocyte come at the site of inflammation still later? Why is so? Let me tell you. Why neutrophils are the first responder to the site of inflammation? You must know 4 reasons. In a previous lecture, I have already explained in big detail answer to this question, right? But right now, I will just enumerate these 4 reasons. Why neutrophils are first to arrive? Answer number 1. Reason number 1. Neutrophils are the most abundant or most numerous white blood cells. In our circulatory system, 50 to 70% white blood cells are neutrophils. As they're most abundant, they will reach in big numbers, first of all. Secondly, neutrophils are the most mobile white blood cells. Their mobility is very fast. You know, neutrophils can move very fast because their ability to make pseudopores through which they move, amoeboid movement, right, which depends on cytoskeleton, especially actin and myosin filaments, right? These cytoskeleton of neutrophil is very well developed for rapid locomotion of movement as compared to the cytoskeleton in other white blood cells, right? Here I would love to mention one thing. You know, when neutrophils are moving towards a target, the front end of the neutrophil is having mostly actin filaments, and rear end of the neutrophil, back end of the neutrophil, is having mostly myosin filaments. If you imagine neutrophil is like a car moving, rapid moving cells, neutrophil is like a car, then front wheels are driven by actin filaments and rear wheels are driven by myosin filaments. These are molecular system which moves the neutrophils, and it is most developed in neutrophil as compared to other white blood cells. So we can say now we have 2 reasons why neutrophils are first to reach at the site of inflammation. Number 1, they are most numerous among the white blood cells in circulation. Number 2, they are most mobile as compared to the other leukocytes. Number 3, when there is pyogenic organism present, the chemical mediators, especially chemokines, which attract the white blood cells, those chemokines and those chemoattractant, which specially attract the neutrophils, are produced first of all and in highest concentration. Let me explain it. Third reason why neutrophils are first to reach at the site of inflammation is that at the site of injury, the chemical mediators of inflammation which are produced, among them the chemoattractant, right? Chemoattractant mean which chemoattractant which specially attract the neutrophils. They are produced first of all. It means the call to neutrophil is first of all and most loud call, because these chemical mediators which attract the neutrophils are not only produced first of all, they are produced in big numbers, high concentration. For example, production of human necrotic factor, interleukin-1, but more importantly, we should talk about leukotriene B4, C4, especially leukotriene B4, complement component C5a, and as I mentioned previously, most powerful chemotactic agent for the neutrophil is interleukin 8. then even bacteria release far more a special type of bacterial products and peptides which attract the neutrophil specially, right? So, and then platelet activating factor, which is produced at the site of inflammation. So, all these things that leukotriene D4 and interleukin 8 or complement factor C5a, or there is what I mentioned, bacterial peptides or platelet activating factor, all different factors. These are very powerful chemoattractants, especially for neutrophils. So that is one more reason why neutrophils are first to reach at the site of inflammation. Now we have three reasons. Yes, number one, neutrophils are most numerous in circulation, so they reach first of all. Number two, they're most mobile among the white blood cells, so they reach first of all. Number three, the calls to the neutrophils are very early and very loud. The chemoattractant for neutrophils are produced very early after injury and in high concentration. So neutrophils are first to reach. And then they keep on accumulating at least for 48 hours. 24 to 48 hours, they keep on accumulating. Why? Because the first wave of neutrophil reaches to the focus of injury, first wave of neutrophil reaches focus of injury due to chemoattractant which were produced by the resident and local injured cells, from chemoattractant which were produced by the mast cells or local resident macrophages or endothelial cell or other injured cells or bacterial peptide. But once first wave of neutrophil arrive, then those neutrophil also produce more chemotactic factors which attract the second wave of neutrophil. Then a second wave of neutrophil will arrive. Then first wave and second wave of neutrophil will produce additional chemotactic agents for more neutrophils, and third wave of neutrophils will come. What I'm saying, as money begets money, neutrophil begets neutrophils. It's just like that. Neutrophils are accumulating at the site of injury and contributing to the pus, and the neutrophils which have already reached there, they are calling more and more neutrophils in their chemical language by chemotactic agents. So these are four reasons why neutrophils are first to reach at the site of injury, because they are most numerous, because they are most mobile, because chemotactic agent for neutrophils are produced first of all in highest concentration, and once neutrophil arrive, they produce the chemoattractant which attract more and more neutrophils, right? So we have discussed two things. Number one, what is pus, right? It was very simple. That pus is opaque, creamy, viscous material which is present at the site of liquefactive necrosis, where there is necrosis and liquefaction has occurred, local, right? And this consists of alive, dead, and dying local cells, alive, dead, and dying neutrophils, and some other white cells, alive, dead, and dying pyogenic organisms, if they are the cause of pus formation, and all these things are present in protein-rich exudate, right? Now, Mr. Ahmed has asked another question. He asked me that in clinical practice, he has seen that in different patient, he came across pus with different shades and different colors. What is the clinical significance of this? For example, pus can be yellow color. You might come across yellow pus or yellow green pus or bluish green pus or brown pus or black pus, sometimes even red or pink colored pus, or even in clinical practice you might come across pus with yellow granules or sulfur granules. Now, what is the clinical significance of different shades and colors of pus? That I will be discussing in the next lecture, that how the color of pus might give you a clue to the underlying organism. See you later. Best of luck.
Thank you, sir. Thank you very much.